Hi, I'm Dr. Mashur. Welcome to my class. Today, in secondary treatment of wastewater, I would like to discuss aerobic treatment of wastewater. Aerobic treatment of wastewater means the degradation of waste material, or that means organic materials carried out by in the presence of oxygen. And there are different types of methods were used in aerobic treatment of wastewater. And we already thought that there is two studies there in wastewater treatment. That is, one is attached growth system and another one is a suspended growth system. And sometimes there is some techniques were used in the combined form. That means in that process there is involved attached growth system and also in the suspended growth system. Here you can see some of the techniques used in the aerobic treatment of wastewater, and we will discuss each process separately. Now we can see the principle of aerobic treatment. Aerobic means in the presence of oxygen. That means this degradation will occur in the presence of oxygen, and the microbes involved in this aerobic treatment, that is known as aerobes. The principle of aerobic treatment is this microorganism will utilize molecular or free oxygen to degrade organic materials, and it convert these organic materials into carbon dioxide, water, energy and biomass. This is the principle of this aerobic treatment. And in aerobiosis, these aerobes will work individually to convert these organic molecules. In this figure, you can see the principle of aerobic treatment. That is this bacteria or microorganism using organic material, oxygen and nutrients, and it will convert into water, carbon dioxide, energy and biomass. This is the principle of aerobic treatment of wastewater. Now we can see the different methods used in aerobic treatment in separately. First we can see the activated sludge process. The activated sludge process is one of the modern technique, and that is a widely used techniques for the secondary treatment of wastewater, and it's used in a suspended growth system in the activated sludge process. And in this method, this microorganism will use organic materials and produce water and carbon dioxide, and the microbes also use these organic materials to produce new cells. The essential unit of this activated sludge process is it having one aeration tank and it also having one settling tank. In this diagram you can see the structure of activated sludge, and it is having one aeration tank and it is also having one settling tank. Now we can see the working of activated sludge process. The primary treatment effluent will enter the aeration tank of the activated sludge, and in that wastewater will kept for 6 to 10 hours for the aeration, and this aeration will encourage the growth of microorganism, and it also promote the conversion of organic materials to water and carbon dioxide. That means this aeration will promote the growth of organism, and it also encourage the oxidation of these organic molecules. And the biomass formed in this activated sludge process will be flocculant that contain microorganism, and that will settle in settling tank as a sludge. A part of this sludge will be recycled into aeration tank as a seed of microorganism to increase the number of microorganism in aeration tank to enhance the decomposition of organic materials. That's why this process is called as an activated sludge process. That means a part of this sludge will be recycled to aeration tank to increase the number of bacteria present in the aeration tank for the decomposition. That's why we can enhance the decomposition of organic material. That's why this process is called as a activated sludge process. And the wastewater in the aeration tank with a return sludge mixed with a high amount of bacteria, that high amount of microorganism, that is called as a mixed liquor. Different types of microorganisms were involved in this activated sludge process, like bacteria, protozoa, rotifers, and like organisms involved in the activated sludge process. And the majority of bacteria involved in the active cell process will be heterophilic, which includes achromobacter, alkaligens, arthrobacter, flavobacterium, pseudomonas, and zooglea, etc. Now we can see the advantages of activated search process. Compared to biological filters, activated search process needs only less space and have a low cost and is having efficient treatment system, and excess studies has a higher fertilizer value, and it has some disadvantages as it is having reduced high rate of power and it is a time consuming process. This is about the activated search process. In aerobic treatment of wastewater, the second method is oxidation ponds or stabilization ponds. Oxidation ponds or lagoons are the secondary wastewater treatment methods mainly used in rural areas, and this oxidation ponds are comes under suspended growth system. This is also known as facultative ponds, which use both aerobic and anaerobic process. These are large ponds with 1 to 2.5 meter depth, and this pond having mainly three zones. One is aerated zone, and second one is middle facultative zone, and the bottom anaerobic zone. And the oxidation in the stabilization or oxidation ponds is mainly carried out by bacteria or algae, and the microbes involved in oxidation ponds will be aerobic, anaerobic, and facultative aerobic. And in this figure you can see the structure of oxidation pond, that you can see aerated zone, facultative zone, and anaerobic zone. Now we can see the process of oxidation pond. The algae present in the upper layer of oxidation pond will undergo for photosynthesis and it will release oxygen. Chlamydomonas, euglena, chlorella centesimus are the common algae found in the oxidation ponds. And this algae is involved in nutrient uptake, mainly nitrogen and phosphorus. And some algae can able to fix nitrogen. And we already told that this algae will perform photosynthesis, and this photosynthesis will lead to the increased pH. At the increased pH, this phosphate will precipitate as a calcium phosphate, and the NH4 ions lost as a NH3. And we told that during the photosynthesis, this algae will produce oxygen, and this oxygen will be utilized by heterophilic bacteria present in this pond. In oxidation pond, wind will induce the mixing of air with water, and it also helps to maintain aerobic condition in the upper layer of this oxidation pond, and it also helps to for the exchange of nutrients and gases between autotrophs and heterotrophs present in this oxidation ponds. And this oxidation ponds also contain some photosynthetic bacteria, and this bacteria will utilize hydrogen sulfates, and that will helps to remove the odor due to this hydrogen sulfates. In oxidation ponds, bacterial heterotrophs are the principal agent that is responsible for the organic degradation. This heterophilic, aerobic, and facultative aerobic bacteria will utilize oxygen that is produced by the algae during the time of the photosynthesis to degrade organic molecules to carbon dioxide and micronutrients, and that will be used by algae. And the dead bacterial and algal and other organic salts will be settled down in the bottom of the pond, where the anaerobic bacteria will do the anaerobic digestion. Anaerobic activity will produce the organic acid, and later it will be converted into hydrogen sulfate, methane, carbon dioxide, etc. And the H2S produced by this anaerobic bacteria will enhance the growth of some photosynthetic bacteria found under the layer of algal cells. Purple sulfur bacteria is one of the common photosynthetic bacteria found in the photosynthetic ponds. Some of the carbon will lose as a carbon dioxide and methane into atmosphere, and most of the part of this carbon will be converted into the biomass. And this oxidation pond has some advantages. One is this oxidation pond is economical and there is no need of a skilled person for this operation of this oxidation ponds. And it has some disadvantages also. Sometimes unpleasant odor and mosquito breeding also may occur in this oxidation ponds. And for this oxidation ponds we need a large area. This is the oxidation pond. In aerobic treatment, the next method is trickling filters. Trickling filters is also known as percolating filters or sprinkling filters. And it is an example of fixed film sewage treatment method. And this treatment method is commonly used for domestic sewage treatment and also for industrial wastewater treatment. Now we can see the structure of a trickling filters. And this trickling filter having mainly four major components, and first one is having a tank, second it having a wastewater distributor, third it having an under-drain system, and fourth one it having a clarifier or settling tank. We can see the tank of trickling filter. It is a rectangular or circular tank. It's containing medium with a depth of 1 to 1.25 meter, and the medium used in trickling filter is stone, ceramic materials, metals, etc. And this media provide large area for the microbes for growth and to form biofilms. And the next part is the wastewater distributor. And the role of this wastewater distributor is to provide uniform water load over the filter medium. And this wastewater distributor have a central shaft that will rotate with sprinklers to sprinkle wastewater over the filter medium. And the next part is the under-drain system. This under-drain system helps to collect the treated water from the trickling filters, and it also collect the biomass that is left from the biofilms of this medium. Next is a clarifier. It is also called humus tank. It helps to separate solid from the treated water. Now we can see the process involved in the trickling filters for the degradation. The biofilm formed of the medium or the bed is responsible for the degradation of organic material present in the wastewater, and this biofilms is known as zooglial films, and these biofilms contain number of organisms like bacteria, protozoa, fungi, algae, etc. An extracellular polymer named glycocalyx is essential for the bacteria to anchor with this filter material or filter medium, and mainly zooglial bacteria is responsible for the synthesis of this glycocalyx. While the progression of the degradation in the trickling filter, the thickness of biofilm will be increased due to the absorption of more and more microorganisms on this filter medium. The biofilm thickness will lead to the limited oxygen diffuse into the inner part of the biofilm, and that's why it's created an anaerobic environment in the inner part of this biofilm, and it also reduces the supply of organic materials into the inner part of this biofilms. Due to the limited oxygen supply and also reduced organic supply to the inner part of the thickened biofilm, this biofilm will start to slough from this material, and new biofilm will be start to form over this filter medium. Bacteria include Zooglea, Pseudomonas, Pavobacterium. Some other nitrifying bacteria were also present in this biofilm. Fungus includes Fusarium, Penicillin, Mucor, Aspergillus yeast are the major fungi found in this biofilm of this trickling filters. And algae like Eulothrix, chlorella are the major algae found in that biofilm of trickling filters. Here we have seen that the biofilms. Biofilms are the group of microorganisms in which cells are stick on each other on a surface, that is known as a biofilms. And these cells are frequently embedded within a self-produced matrix of extracellular polymeric compound. Advantages of trickling filters are easy operation, low maintenance cost, and its reliability. And the disadvantage of this trickling filter is overload of organic materials will lead to clogging of this filter, and this clogging will lead to excessive growth of bacteria, and that will cause the odor from this trickling filters. This is the trickling filters. And in aerobic treatment of wastewater, next method is a rotating biological contactor. Rotating biological contactor or RBC is a fixed film method of a wastewater treatment, and this medium used in this RBC will be in moving. And this RBCs consist of series of closely spaced discs which are mounted on a horizontal shaft, and this disc were submerged 40 to 60% in water and it rotates slowly. And the microbial growth attached on the surface of the disc and form biofilm with a thickness of 1 to 3 millimeter. And the rotation of this disc will provide the aeration to the biofilm formed on this disc. Increased rotation improves the oxygen transfer and enhance the contact between attached biomass and water. Bacteria like D-sulfovibrio, Bigiotova, Nocardia were present in the biofilms. Protozoa, metazoa were also present in this biofilm of this rotating biological contactor. And algae like Oscillatoria were also present in this biofilm of this rotating biological contactor. In this figure, you can see the structure of a rotating biological contactor. It has a series of closely spaced disc it is 40 to 60% submerged in this wastewater and is arranged in a horizontal shaft, and it has one motor to rotate this disc. This is a structure of rotating biological contactor. Now we can see the biofilms formed over the RBC's medium. And this biofilm formed on the RBC media having mainly two layers. One is aerobic zone that will be whitish in color, and second one is the anaerobic zone that will be in blackish in color. And the anaerobic zone that will be in blackish color is due to the precipitation of ferrous sulfate. When ferrous reactive with hydrogen sulfate produced by disulfovibrio, and organic acids, alcohol produced by other microorganisms will be used by disulfovibrio. And the aerobic zone that will be in whitish color, and the HDS produced in the anaerobic zone that will be diffused into the aerobic zone, and bacteria Bigiotova use this hydrogen sulfate, and elemental sulfur will be deposited in the bacterial cell. Now we can see the advantage of rotating biological contactor. It's only need short residence time. It needs only less space, and this RBC having high BOD removal capacity and have some disadvantages also. The dispersal of sludge formed in this RBC is the major problem, and sometime odor will be formed from this RBC, and this RBC operation will require a skilled person. This is the rotating biological contactor. In the aerobic method, next is the fluidized bed biofilm reactor. This is also known as fluidized bed reactor. Is the one of the recent method used for the aerobic treatment of wastewater. And this method is a combination of attached growth system and suspended growth system. And the biofilm formed on solid medium consists of small particles, and these particles will be maintained as suspension due to the upward flow of wastewater into the reactor. And the microorganisms present in the biofilm that will degrade the organic molecules present in the wastewater. Now come to the advantages of this fluidized bed reactor. It is suitable for the treatment of high strength industrial waste, compact small reactors, space saved, and produce only low sludge, and it needs only low capital and low operational cost. This is the fluidized bed reactor. These are the major aerobic treatment used for this wastewater treatment. Some other methods also used for this wastewater treatment, and these are the major methods. That's why I explained in this class. I hope you understood this class. Thank you for the listening.